Hello everyone, it's my great pleasure to give a presentation here at the Africa GloMiNe. And just to introduce myself, I'm assistant professor at the University of Lausanne, and I'm also a principal investigator in the NCCR Microbiomes. And today I'm going to talk to you about undernutrition and the microbiota in Africa, so really bringing to the forefront a subject actually that is important to Africa and is also tightly linked with the microbiome. So let me first introduce what I mean by undernutrition, as there often also some confusion about it. Undernutrition actually comes in several flavors. We have stunting, which is a reflection of underlying chronic undernutrition, and basically means that children are too small for their age. And there's a second form of undernutrition, which is wasting, sometimes also called acute undernutrition, which means that children are too thin. And while often we think about wasting when we think about undernutrition, stunting is actually much more common and affects roughly 1/4 of all children under the age of 5 years globally, many more actually in some African countries. And it's thus about 3 times more prevalent than wasting and also childhood overweight that are both affecting roughly 7 to 8% of children under the age of 5. Undernutrition has very severe long-term consequences. They include higher morbidity, also diminished cognitive development. Further, children that are undernourished in early life have also higher risk of obesity and cardiovascular disease in later life. And second, and also most importantly, there's also intergenerational effect. So basically mothers that are stunted are also having a higher risk of having stunted children themselves. So what is now the etiology of stunting? Obviously, it has a lot to do with nutrition, especially a nutrition that is poor in macro and micronutrients, also restricted in calories, and exposes the children to different toxins, such as an example, aflatoxins. Then there's also a vicious cycle that takes place between infection and undernutrition, by infection worsening actually the undernutrition state, and undernutrition being a favorable ground for more, especially like enteric infections. And in recent years, another third entity has also come back on the agenda, which is changes to the gut ecosystem, and is sometimes also called environmental enteric disorder, and is characterized by a shortening of the villi in the small intestine, increased permeability, and also chronic inflammation. And this syndrome is also accompanied by changes to the microbiome, as we will see in the further talk. So it's then believed that all of these 3 entities are contributing independently to stunting and also to the associated immunological and neurodevelopmental consequences. However, for the moment, it still remains a matter of debate how these different entities are linked, if they all contribute in every case to stunting, or how this really is happening in every single child. So the big question was then obviously, is there a role for the microbiome in undernutrition? So is the microbiome causally having a role in it? And it's now almost 10 years already that a very beautiful study has come out from the group of Jeffrey Gordon in the United States, where they made a first proof of principle that the microbiome really plays a role. And what they did is that they took fecal samples from either healthy children, so depicted here in green, or from undernourished children, here actually depicted in red, and they transplanted them into germ-free mice, so basically in mice that don't have their own microbiota, and they exposed them, both of these groups of mice, to what they call a Malawi diet, which is a diet that is deprived actually of macro and micronutrients. And as depicted here, the group that had the normal microbiota remained normal, while the group that actually had the microbiota from the undernourished child was losing weight. They then re-nourished both groups of mice and could see that basically the mice that were formerly undernourished were gaining also again weight. However, when they switched them back on the Malawi diet, the mice that had the microbiota from the undernourished child were again actually like losing weight. And this was really a beautiful demonstration that the fecal microbiota is directly and also causally related to weight loss and also to impaired growth. So a second study that came out then a few years later started to address also the question, what could be basically the causes and what could be done. And what the group did is that they co-housed mice that either had the microbiota from the healthy or from the undernourished child in the same cage. And now mice have this peculiar habit that they actually eat each other droppings, meaning that they have basically like a constant exchange also of their microbiome. And what the researchers observed was when they had actually mice that had an undernourished microbiome in the same cage with mice that had a healthy microbiome, this formerly undernourished mice were doing much better in their growth. And this really said that there are indeed specific missing taxa in these undernourished mice, that they can come actually from outside, as an example, through the feces from another mouse, and are then also able to ameliorate the growth of the latter. However, today this still remains somehow elusive what the molecular mechanisms beyond this really are. So going further, there was then another study looking at the succession of the microbiome in early life. As we heard in the first talk, the microbiome is normally acquired at birth. It then goes through different stages similar to an ecological succession. And this succession is actually always taking place more or less in the same way. Now, when one looked at the microbiota of undernourished children, it was visible that basically this succession was disturbed, and basically these children didn't only have a delay in their anthropological growth, but they also had a delay in the microbiota that they were displaying. So basically the microbiota of a child of, as an example, 18 months looked much more like the microbiota of a healthy child of 14 or 15 months. Further, there was also research looking beyond actually this initial succession, so when the microbiota is already more or less stable. And there also our own research in Central African Republic and in Madagascar came into play. And what we could show was that there was a very specific microbial signature in these undernourished children, and there was especially a very high load of bacteria in the small intestine that was surprisingly dominated actually by bacteria that normally live in the oropharyngeal tract. So almost 60% of these bacteria were basically bacteria that were ectopically colonizing from the oral space. We then also try to find out what they do, and we could show, as well as others also in the field, that these bacteria seem to be associated with inflammation. So here you have data that shows actually cytokines in the small intestine of children in Madagascar and the Central African Republic. And we compared the cytokines between duodenal samples from children that either have this bacterial overgrowth in the small intestine or not. And as you can appreciate, there's a whole panel of so-called pro-inflammatory cytokines that are more abundant in children that have this small intestinal bacterial overgrowth with oral bacteria compared to the ones that don't display it. We then also went on and assessed if there are other pathophysiological mechanisms that are disturbed by this extreme presence of bacteria. And here I show you data that we produce in vitro with isolated strains from these children, so strains that we isolated from the small intestine. And as you can see here, we have the control in black, then a positive control in white, where we use Lactobacillus paracasei, which is a probiotic, as a control actually that did not affect the cells, and Escherichia coli, that is a bacterium that we know is decreasing lipid absorption. And you have here on the y-axis actually the absorption of oleic acid into the cells. And to our big surprise, all of the oral bacteria actually also led to different degrees to decrease lipid absorption, meaning that these oral bacteria don't only lead to inflammation, but apparently also to a decrease in lipid absorption in epithelial cells. So the big question was then obviously, so is this also happening in maybe a more complex system? To this purpose, we then went on with one of the strains indicated here in purple, which had the biggest effect in vitro. And we also assessed for lipid uptake in a mouse model. And as you can see here, again, the control mice, then Streptococcus salivarius treated mice indicated in blue, and Lactobacillus paracasei, the control treated mice in green. There was indeed a significant decrease in lipids that were absorbed into the small intestine of these mice when the mice were exposed to Streptococcus salivarius. And the same was also the case if we looked more systemically, so in the liver, where we could again see that mice that were basically treated with Streptococcus salivarius had also lower levels of lipids that were taken up into the liver. So there was really a clear role of this oral bacteria in lipid absorption, and thus also a direct role between the microbiome and the pathophysiology underlying stunted growth. So to summarize the research that has been done since the very first study I showed you in the beginning, I think we and others in the community could show that there's really a community homogenization with oral bacteria suddenly being in very high numbers in the small intestine, where they lead to chronic inflammation and also decrease lipid absorption. And then we also saw, and this I didn't show you yet, a decrease actually in different bacteria that are important for butyrate production and also bile conversion in the feces, and also very high presence of enteropathogens and pathobionts. So obviously now the question is, what can be done? And the answer is that we should try actually to revert the microbiome back to a healthy state. This can be done with so-called microbiota targeted interventions that can take different flavors, antibiotics, but we just heard this is probably not the way forward. Prebiotics, probiotics, or symbiotics, which are fibers, living bacteria, or a combination thereof. Also postbiotics, which is basically meaning that you provide bacterial metabolites that might be missing, fecal transfer, or also phage therapy. And there has been one approach that was especially promising, which was basically modulating the microbiota through nutrition. It's again the group of Jeffrey Gordon that did so in a study that has been published now 2 years ago, and where they looked at the microbiota composition if they gave specific diets to the children. So here you see the trajectory in such a PCOA of a severely undernourished child, and you see that even if it's treated with the standard care 12 months after discharge, the microbiome is still really different from a healthy subject, while when they were treated with different dietary foods, the microbiome of a moderately undernourished child could be reverted in the case of a food that is called MDCF2, almost back to the composition of a healthy state. And right now, there are first clinical trials in humans ongoing, and I'm very excited actually to see if this also holds true in a larger data set, and also if this can revert the actual stunting phenotype. So in summary, I hope I could show you that there are specific microbial signatures that are directly implicated in child undernutrition, that microbiota targeted interventions could be a promising tool to enhance the treatment of undernutrition. And finally, I think it's really important to note that in the last 10 years, a lot has happened in the field, but nevertheless, there's still a lot that has to be researched in this field to really have good treatments for these children. And therefore, there's really more research that is needed. And with this, I would like to thank everyone that was implicated in this research. I would also like to thank you for your attention, and I'm happy to take any questions that you might have.
Thank you very much, Pascale, for this great talk. We would have time for about 1 or 2 questions. I don't currently see one in the chat. Please, if there's anything urgent, don't hesitate to put it in there. Otherwise, perhaps from my side, Pascale, I was wondering, clearly in your preclinical studies, having somewhat of standardized microbiomes as well as standardized diets for the mouse experiments may be key going forward to also compare results across different geographies. Do you know what the status is there in terms of having such standardized protocols and diets and microbiomes available?
So I think for the moment, there is not really standard microbiomes available. So basically, there's only now that people start also to realize that the microbiome is very different. So that basically, like, you also have to look in different countries. I know that in the group of Jeffrey Gordon, they really try to have like a pipeline where they also use this germ-free piglets as an example to transplant the microbiome and then test it with different diets. It's still very in its infancy, I would say, but it's very exciting actually, like how people start to standardize to really have a better impression of what is happening.